a live movie, but you can see what's going on here. You can see what this constant pulling and, let me do that again, the constant pulling and pushing forces that are going on, that these are the chromosomes going back and forth, back and forth, back and forth as the cell's getting towards metaphase. So it's constantly sliding them back and forth in order to be ready to go into the next stage, which is anaphase. So it's really important for the cell not to go to actually go to anaphase until everything's lined up, right? Or until the chromosomes are essentially in the middle. Because remember what I said, it is so important that we get half the chromosomes on one side, half the other. So even if we have 95% of the chromosomes in the middle, but one duplicated one is way over on the other side, that's going to be a disaster because we'll get both chromosomes in one cell, a daughter cell, and not in the other. And so this is where a very important mechanism comes in that regulates this checkpoint, the metaphase to anaphase checkpoint. And what it does is it's sensing, exactly how this works is still under debate, but what it's doing is sensing whether there's equal tension at the kinetochores, because when the chromosomes are roughly in the middle, there'll be basically tension on both sides of the kinetochore, because the kinetochore will be in the middle, being essentially pulled by both sides. Now, when there is not tension at the kinetochore, this protein here called CDC20 is going to be repressed. But when the tension is equal, CDC20 now becomes available, and it will bind a set of proteins called the anaphase promoting complex, or APC. APC is a ubiquitin ligase. Remember how I told you that ubiquitin ligases could be activated by another protein binding? Well, that's what happens here. So the binding of CDC20 is going to activate this ubiquitin ligase complex. Now what happens in G2 is that all the chromosomes, remember they're duplicated, are held together by a sticky glue that keeps them together. And that's called cohesin. It's basically glue that's holding them together. So even when we get to metaphase, okay, after that M phase CDK becomes active, there's no chromosome, the kinetochores are attached. There's pulling forces on each side, but if you ask, well, why aren't the duplicated chromosomes going apart? It's because there's the sticky glue. So even though there's pulling on both sides, the glue is keeping these things together. So in order to go from metaphase to anaphase, we have to get rid of the glue, right? Otherwise, they can't separate. So that's where this ligase comes in. This is where the key checkpoint comes in. What the ligase, the APC with its CDC20 bound to it does is it ubiquitinates, remember that, and degrades a protein called securin. And securin is bound to a protease, or a pair of scissors essentially, called separase. So throughout the cell cycle, securin is keeping the protease inactive. When APC becomes active, the ligase becomes active because there's tension on both sides. Now what happens is securin's degraded, separase becomes active, it goes into the cell, it cuts all those, all the glue parts of things, and now the chromosomes can move together, apart, okay? So that's the metaphase to anaphase, all regulated by this checkpoint, which is the tension regulator. So that's how we go from metaphase to anaphase. Now, in addition to that, this is another view of the same thing. So one of the key things that the ligase does is it causes the degradation of the securin. But it also causes the degradation of the cyclin. So here, again, if there's a lack of tension at the kinetochores, CDC20 is not active. When there's tension on both sides, CDC20, as I just told you, binds the anaphase promoting complex and causes it to be an active ligase. In addition to hitting securin, the other thing it does is it causes the degradation of cyclin by ubiquitinating cyclin, as shown here, causing it to be degraded. So that's how we get out of M phase. So this tension checkpoint is not only regulating anaphase, metaphase to anaphase, but it's also regulating the end of M phase. And the key thing is that the securin is degraded rapidly. So we go metaphase to anaphase. The cyclin is more slowly degraded so that it will mark the end of M phase, but it's going to take more time before M phase ends. So the immediate effect is the metaphase to anaphase transition, and then the more longer time effect is the eventual ending of M phase, because remember, once cyclin is gone, the MCDK is inactive. Okay, so the checkpoint is regulating both steps. So what is anaphase? Well, I told you that anaphase is essentially we're going from metaphase to the anaphase. The chromosomes are going apart, and anaphase is composed of two events. They're called anaphase A and B, but it's not like A happens and then B happens. A and B happen together, but people have found it easier to describe them as separate events. So that's the way I'll do it. It's easier. I agree. So the anaphase A is what's happening to the is action that's happening at the kinetochores. So remember the kinetochores. I told you the NDC80 is grabbing onto the end of the microtubules, and I said that they're not actually glued on there. They're just sort of held on. When the glue is cut, the cohesin is cut, now what can happen is that the NDC80 will keep moving towards the minus end. And the reason that happens is it's not that the NDC80 is a motor, but that the plus ends will start depolymerizing. Okay? So this is what's happening. So again, here's the centrosome over here. Here's the kinetochore. Chromosome would be right here. So what happens is that the plus ends will start disassembling. Now, actually, if you remember that movie where it was going backwards and forwards, what was happening was that was actually due to polymerization and depolymerization at the kinetochore. So when it would move in one direction, you'd get polymerization on one side, depolymerization on the other. When it moved back, you'd get depolymerization on one side, polymerization on the other. So it's going backwards and forwards at metaphase like this through alternate polymerization and depolymerization. Once the cohesin is cut, then we get this rapid depolymerization here, and that the NDC80 is not a motor, but it just keeps trying to hang on for dear life to whatever is left of the microtubule. And I have looked for a long time to find a movie that illustrates that, and one of a past 440 student helped me find it. So it wasn't easy to find. Let's see if I can find it. Here we go. This is it. So this is what it looks like. So here we have, this is the microtubule. Here's the kinetochore, and this is a really good example. Right? Okay, so that's exactly what is happening in anaphase A, is that the microtubule keeps polymerizing, and the NDC80 complexes right here keep grabbing on to whatever is left. Now, anaphase B is similar, or it also uses microtubules, but it's a somewhat different kind of motion, and it actually involves two things. So anaphase A is the movement of the chromosomes towards the poles. Anaphase B is the movement of the centrosomes apart. Okay, so think about it. It makes a lot of sense. If the chromosomes are moving towards the kinetochores and the kinetochores themselves are moving apart, that's going to cause the DNA to get further and further apart. So in anaphase B, these centrosomes are going to move toward the edges of the cells. And again, this involves microtubules. So anaphase A, remember, involved the kinetochore microtubules. Anaphase B will involve the remaining two sets. So one of the things that happens at anaphase is that there are dynein motors that will be embedded in the membrane. Now remember, dynein, they're ones that are motors that go towards the minus end of microtubules. So what happens is these dynein motors will grab onto the astral microtubules, the ones heading out toward the edges. But because they're embedded in the membrane, these dynein motors, these special ones, they can't move. So what happens is, instead, as they try to move towards the minus end, they're essentially pulling the kinetochore towards the centrosome toward them, right? They're trying to move, but they're held in the place. So if I'm a motor and I'm stuck here and I'm grabbing onto a rope, I'm just going to keep pulling it towards me. Okay, so part of anaphase B is the dynein motors grabbing hold of the microtubules and literally pulling the centrosome toward them. That's the second class. The third class involves the interpolars. And this one's harder. I'll try to explain it. It's slightly confusing. But I told you that the interpolars you saw in the Alberts were connected together. There were those little black dots that were holding them together. I didn't tell you what they were, but they're a special class of kinesin molecules. Remember kinesin? They're a special class of kinesin molecules that have two heads, as shown like this. Okay, so this is what they look like. Now, what does kinesin do? Kinesin wants to go out to move towards the plus end. Okay? Dynein wants to go towards the minus. Kinesin wants to go towards the plus. And so during the first parts of the M phase up till anaphase, these things are just sitting there holding the interpolars together. But what happens is that at anaphase, each of these heads is going to start moving out to the very end of the plus end. And this is where it gets slightly confusing, because as they move, they're also going to cause the plus ends to grow. So let's look at that in more detail. So here's the plus end. Here's the plus end. The interpolars are going in both directions. So what's going to happen is that, remember, plus ends, microtubules can grow and shrink. So what will happen is you'll get a bit of growth here, let's say at that plus end, and then this kinesin head will move out towards it. And you get a bit more growth, and it grows out towards it. So the reason it doesn't shrink is because the kinesin head keeps moving out to the newly growing plus end. Okay, so remember then, grow, and I'll show you a little animation of this in just a second. Grow, add. So if you imagine now that this one is moving towards a plus end that keeps increasing, and this one is moving towards a plus end increasing, because the distance between the heads remains fixed, what it's doing is basically pushing the microtubules that are growing longer in the middle out. Okay, so it's pushing them out. So anaphase B involves two components. There's a pulling force from the dynein out here, pulling it in, and a pushing force from the middle as those heads cause the microtubules to extend. So I wish Alberts had a movie of this, but I made my own little animation. You can see if this is helpful. So again, let's just imagine here are the two centrosomes. Here's the microtubules. There's the kinesin. And just imagine, so you get growth, and then it slides, right? Get growth, and then it slides. Let's try that, see if it does that again. Right? Growth, slide, growth, and then it would slide. And just if it keeps doing that over and over again, what it's doing is pushing these two things further and further apart. Okay? So that's anaphase B. So we've talked about the microtubules. I want to now come back to, because there's a type of intermediate filament that plays a key role also in this process. And these are called the intermediate filaments. So we've talked about the nuclear envelope, remember, that goes around the cell. And here's the nuclear pores, you remember those. What I didn't tell you is that underneath the nuclear envelope is a type of intermediate filament called the lamins. And those are important because they give strength to the nuclear envelope. They're a network that underlies the membrane that gives it strength. Now, what has to happen in M phase is the nuclear envelope has to break down. But what's opposing that breaking down is this meshwork of intermediate filaments. So one of the key phosphorylation events that occurs as we go in, from G2 into M is phosphorylation of the lamins by the M-CDK complex. And what happens, that phosphorylation causes that strong network to break down. So underneath the nucleus, instead of this really rigid thing that's giving the nucleus a lot of strength, because of the phosphorylation, the whole network falls apart. And now the nuclear envelope doesn't have anything really strong, which, as I'll show you in a second, allows it to be broken apart. So that's the condition that will occur through most of M phase. At the end of M phase, when cyclin is degraded, so we no longer have an active cyclin CDK complex, what will happen is that this, the lamins, which are no longer being phosphorylated, will be dephosphorylated by phosphatases. And when they're dephosphorylated, they can begin to reform. And it's actually the reforming of that network that starts bringing all these little pieces of nuclear envelope that were all little fragments throughout the cell. It will cause them to reassemble until eventually we get a nucleus. Okay, so you can see that's a really clear case where we use that MCDK complex. Now it turns out Alberts makes it look as though once you dephosphorylate, the nuclear envelope just falls apart. But that didn't actually ever make any sense to me, because even though the nucleus was weak, it's still it's still membrane. It's not just going to spontaneously fall apart. And one of the things that was discovered recently was that there's actually a ripping force that actually has to rip the nuclear envelope open. So what happens is, first we have the lamins under the nuclear envelope, and they're phosphorylated, so they're no longer strong, but we still have a membrane all around the nucleus. So here are the two centrosomes, and one of the interesting things that happens is, right as we're going into prometaphase, remember dynein? There's some dynein molecules that actually bind to the pore. They grab hold of some microtubules, the plus end of these microtubules coming out from the centrosome, is shown like this, and they start moving toward the plus end. And it's actually, what they're doing is they're moving, they're kind of basically putting some force on the nucleus. Not much, just a little, but that's what causes the nuclear envelope to break apart. Okay, so just that little bit of force, because it no longer has the lamins underneath, is enough to to cause this thing to shred. If the lamins were still there, like in most of the cell cycle, this wouldn't have any effect because it's just a weak force. So the combination of the breakdown of the lamins, the intermediate filaments underneath, as well as the pulling force, is what causes the nuclear envelope to break down. So again, you can see, I think, the way these different filament systems are interacting together. So the final part of this I want to tell you about is the actual physical division of the cell. So we've talked about the role of microtubules, we've talked about the role of intermediate filaments, and now we're going to come to our third component, which is actin, because actin plays a key role in actually physically dividing the cell. And what's going to happen is, as we get in telophase, is that an actin ring is going to form right around the middle of the cell. This is what a picture of a cell where it's being pulled, and you can see the two parts are kind of being pulled in. But there's this actin ring that is right here. And so what happens is it starts like this, and it constricts like this, and eventually it's going to pinch off. And what the key thing that happens in this case with the actin is it's just transient. It's a transient assembly of actin right around the middle. So this is what it looks like sort of on a side view. This would be if we looked at an end view. You'd see all these short actin filaments like this going right around the middle. And what's key is that myosin jumps in here, little molecules of myosin jump on here, and they pull the actin against each other, okay? So they grab hold, and they're motor proteins, and they're pulling the actin together. So what happens is these actin filaments will be pulled against each other, and then they'll depolymerize, so the ring will shrink, and it will also, the actin will get shorter and shorter until eventually, basically, the cell gets pulled in half, okay? So you can see now how that third component comes in. The actin is absolutely critical for this to occur. And I just have a little movie I want to show you of that. So you'll see the actin right there. Did you see that in that one spot? I think there'll be another one where you just see this incredible rapid assembly of actin. Let's see, there should be there you go. Do you see that? Another one. The actin just assembles, pulls, and then goes away. So again, it's this ability of the actin, just like the microtubules, to be incredibly dynamic that allows this to work. Okay, so actually I'm going to end it there for today, and then on Monday we're going to start talking about cancer and the many processes involved in that. And I think you'll really see how a lot of the basic biology we've been talking about during the first 4 weeks is really going to connect through to a cell biological understanding of the processes of cancer.